All right, how you guys doing? Dr. Wolshan was out of here on time because you guys had an exam, whoever was here. How'd that go? Is that all right? All right, so last time, of course, we went over the exam. So if you didn't get that, I actually don't have them with me today, but you can come by my office or let me know when you like to come by my office to get that. Before then, we had started covering the kind of microbiological aspects of wastewater treatment, specifically how do we handle our growth, how do we model it mathematically, so how do we do a mass balance on the growth of microbes. That would give us the information we need in order to do a mass balance on how much food they're consuming, all the substrate, the dissolved organics that we want them to consume and convert into something physical we can remove. How do we tackle that? So we started on this problem. We had BOD leaving a pond, and we derived an equation which I have since recognized we had a small error in it. I can see a couple errors in it. So hopefully it was just the fact that we were being a little quick at the end there, that none of you caught my mistakes. But we're gonna pick back up here. I'll correct a couple things, and then we will solve part C, hopefully with no mistakes, and get our answer there. So any of you happen to see the mistakes yet? You could even just compare with the formula sheet I posted, but I was supposed to factor out the KS here, so that's one thing, right? That was already factored out to here, and then on the denominator, I believe I have pluses when I need minuses here, and so I think what happened in part was this negative I did not translate, right? So that should have been here. We had also, or let's see, hang on just a second. So what I did was I subtracted, or no, I added both of these to the other side. So that's probably not it. And I moved them around, so that would have been, so that part's fine, but I think there's an issue somewhere. So let's say right here, starting from here, I wanted to get all of the S terms by themselves, so I should have subtracted the S and this guy, the theta KD S. So those should have been subtracted, but I had added them here. So that's where it should have been. So subtract and subtract. So that would mean these are negative, negative, and negative here, negative here. So the final equation that you'll have, these should be subtractions. Okay, so sorry about that. I'll post this corrected version of it, and you'll have this one in the formula sheet. One quick reminder, this equation is the mass balance solution solving for S when we did a mass balance on X for the specific scenario of a no recycle system with zero influent concentration mass concentration of bacteria. So it only holds true for that situation. So don't approach all times where you need to find S as using that equation. I'm giving you that equation in case we work with that kind of a problem, so you don't have to go through this derivation, make the mistakes that I made. But okay, does that make sense? Any issues on that? All right. So with that, part B of this problem was asking for us to give an overall efficiency for the removal of this organic matter. This is sometimes we'll have a formula for a specific type of efficiency, like the filter efficiency, and granular filtration was kind of on a water basis. Well, this is just asking very simply or very generically. We don't have any specific specifics otherwise. So this would be what we started with minus what we finished with divided by what we started with. And we can multiply that by 100% to get it to percentage. So in this case, we started with 95 milligrams per liter BOD. We had 6.9 milligrams per liter remaining divided by the 95. I think that came out to 93%-ish. I think it was 92.7 maybe. Just going to double check my notes here. Yeah. Okay, so we've got that. That's kind of the pretty straightforward, easy answer. So then part C asks us, what's the concentration of volatile suspended solids leaving the pond? So that's asking us what is X final, and we'll recall that X in the pond is equal, since this is, we were told it's well mixed, whatever's in the pond at the moment is the same thing that's leaving, right? So last time we talked about how it's a bit counterintuitive, but we solved for X and all the X's were able to cancel so that we could know S. And now that we know S, we could solve for S and fill in the S's to find X. So it's a little counterintuitive, but we're going to do a mass balance on the substrate to tackle this problem. So I will, in the previous slide we had these equations that I wrote up and the values, the parameters there. So I'm going to screen grab those. It'll interrupt the slides for a moment. And then I'll kind of paste that on the next slide so that you'll have that. So we'll just have that there. And sorry I didn't come better prepared for that. So I'll put that here and give you a few minutes to work on that. So go ahead and start working on mass balance on the substrate concentration, aiming to find what concentration of X we have in this solution. These slides will go away for a moment, but I'll have them right back up. All right, so hopefully you had a chance to get yourself acquainted with this part of the problem at least. I went ahead and started it saying 0 is equal to QS naught minus QS plus, in that volume, our rate of substrate utilization. So we can then remember that the substrate utilization rate can be defined as kind of the opposite, right, of the growth rate. So when microbes are growing, sounds like there's an echo. When we have microbes growing, then the substrate is being used for that, right? So it's a negative relationship with the growth rate. It's also, we factor into it the yield coefficient. So the yield coefficient is how much mass of microbes we get per mass of food we feed them. So the substrate utilization rate can be found as negative Rg divided by that yield coefficient, y. So then we can write all that out. The R of g is that mu times x, and we know that mu, so that's mu times x right here, right? But we also know that the mu is the mu max s divided by ks plus s. That was kind of one of the first things we looked at with the growth equations. Okay, so then we can insert this entire piece, now that we have that written, right in here for the substrate utilization rate. So that's kind of what we do there. Putting that in, we get 0 equals QS naught minus QS minus V times X mu max S, all divided by KS plus S times Y. Okay, so we've got all that written out, and now it's kind of time to simplify and solve for X. One thing we can note is we have mu max divided by Y, so we can actually simplify that to the maximum specific yield coefficient that we talked about the other day. So we're going to simplify in part by adding that in. We'll go ahead and divide everything by Q. That'll give us some more simplification and put the term here in theta. So we'll do both of those things and say this is S naught minus S minus theta times X times, I'm taking out that mu max divided by Y, and instead I'm putting in the K, lowercase k, times S, and all of this divided by that KS plus S. Okay, so that's a little simpler. Again, just need to solve for X here. Fortunately, we know S now, right? So this should be a little simpler. So I'm going to add this piece to both sides. So on the left now we'll have theta X, lowercase k S. And remember, there's 3 different Ks we're tracking with these microbe situations. So make sure that you understand which K is which. For example, when we wrote up these parts earlier, the lowercase k here with no subscript, that's our maximum specific yield coefficient. Our uppercase K with a subscript S, that's that half velocity or half the max substrate constant. That's in food units. And then KD is the death rate. Okay, so there's 3 different ones to track here with these equations. This is just the lowercase k with no subscript. It is K then S, so don't confuse that with K subscript S. Okay, so I'm going to move that to the left side, and I'm just going to go ahead and multiply both sides by the KS plus S while I'm at it. So that's on the left, and then on the right will be S naught minus S, and I multiplied both sides by KS plus S. So to get X then, we have all of that divided by theta KS. So we have S naught minus S times Ks plus S divided by theta lowercase k s. All right, so there's our derivation that we can use. Again, this is for a zero order, not zero order, first order system with zero influent X with no recycle, right? It's for this particular system, we can have this condition that we find. So that's the derivation. Now when we solve for X, I think that comes out to be 43 milligrams per liter. Yeah. Milligrams VSS per liter. And there's our answer. All right, how do you feel about that? Okay. So, and I'll note that I'm giving you this derivation for X for this particular system, but it's up to you to remember that that really only applies to this no recycle system, okay? So be warned, that could get you confused if you're trying to apply it to a system that has something other than what we've shown here. But it's, looking at going from like a batch system to a pond type example, that's the next simplest system in a way, right? It's continuously mixed. You've got water flowing through it. So it's relevant to some situations. Okay, so that's it for the first set of slides on our secondary treatment. And now I'd like to take us through what it's like when we recycle the water, or recycle the sludge rather. So in this case, in a practical sense, we really want rapid elimination of S, right? Rapid eating of the microbe food. So we want a lot of microbes in our container that we're doing this reaction in. And so it serves us really well to fill up sludge here and send it back in. Now I'll remind you that the system here is talking about, we say primary effluent. This is from the primary treatment. So there's going to be some sort of a clarifier here that'll also send to the sludge. So that would be primary effluent is the same thing as secondary influent. Okay, so just note that here. So we're going to have some separation, but now that, that might be, all sorts of junk, whatever happens to be in the sewer lines or the wastewater stream, that will settle. Then we send it to this aeration tank. Here we're adding a bunch of air, typically, whatever bioreactor we're using. We're growing lots of microbes. Then we're going to clarify it, collect water with X in the effluent to be approximately 0 in terms of a mass basis. Of course, we will have some microbes escape, right? That's not quite the point here. We're not expecting gravity to make every single microbe, go away. But in terms of the mass flux or the chunks of the microbes, we have approximately 0 if we are operating properly. You'll see as we work through this that if that doesn't happen, then we're no longer operating properly and we have a big problem. Okay, so then in the aeration tank, we have some amount of X, some amount of S. That means that whatever is coming out here, we have S and X. Whatever we have coming out of the clarifier here, we will have the same S, but the X has changed because the clarifier is changing the concentration, right? So we can do a mass balance if we wanted to right here on X and find that 1 input, 2 outputs, but X is only going through the one output. So we could solve for, this Q here. I'm not even going to give a term for it at the moment because I just want to let you know that you could think about it this way compared to the QE, and you'd see that all of the X comes this way and X is none of it goes that way. So you could solve for that with the mass balance on the clarifier. Then you have this X in this line. We could call it X that's heading to waste, and it would be the same concentration as the X that will be recycled, right? So this XR, and I guess they went ahead and labeled it XR here too, but either way, the pipe down here is just splitting in two directions, right? So whatever concentration we achieved, this is not a little clarifier at this juncture, so the mass balance there is very, very simple. The flow splits in two directions, right? That's all that's saying. So we'll say XR equals X in the waste line. Okay, so then we will have some amount of mass of microbes going back to the start of the situation. That gives us the condition that we wanted with high amounts of bacteria in there. Then what we'll notice, if we think about it, the only thing that controls how much bacteria that come back into the system is the flow rate we use in our WAS line, the waste stream for our activated sludge, or the WAS line. So this Q turns out to be quite important. If we want more bacteria in our activated sludge basin, then we decrease the amount we send to waste. If we've got plenty and we're happy with that, we can increase QW. So this is just essentially the solids removal. And so we're going to go through why that's important and how that impacts our system here. Okay, so a few assumptions. Already kind of touched on this. The clarifier we're going to assume to be perfect. So that means XE goes to 0. We're also going to go ahead and assume that this clarifier does not have any meaningful biological activity, meaning we're not giving it air for these microbes to consume, right? If they had plenty of air, then as they go, it would really, we'd have biological activity there in a meaningful manner, and we'd be growing more microbes. So we're going to assume that, in a sense, the volume is 0 here, or at least the reactions are 0 there, right? It's kind of a decent assumption because we're not bubbling air through it, right? And they don't stay there for too long of a time. So we could think about it as V of the clarifier equals 0, or simply the R prime of G, is 0, or whatever. However you want to think about it, we don't actually have to deal with math. This is just an assumption to say, hey, when we do our mass balance, we don't need that volume. So that's just one of the assumptions there. Okay. Then I already was a little ahead of myself, I suppose. One thing we'll notice is that S in the effluent here is going to be the same S as we have here. So this S, this S, this S, and this S, they're all the same, right? Because we have nothing that can change that between these points. So whatever happens to be in there is the same thing that's leaving. That kind of helps us balance things. And within the scope of our overall mass balance that we might want to do with the control volume out here, the dotted line, we notice that if S is in here and S is in this recycle line, we're not really changing anything about S by recycling that. So we kind of end up ignoring the recycle line for a lot of purposes. Okay, so then XR is equal to XW if we were to say this is XW, right? We talked about that. And since we know that XE is 0 out here, we know that we have, and we also know that we have some flow. QE should be large. That's the flow of our wastewater that we're going to send to some receiving water. Right? That's the flow of wastewater that's leaving our system. It's finished, it's cleaned, it's purified wastewater. So in that sense, we know that XW has to be quite a bit bigger than X. So we had some amount of concentration of the bacteria. We separated them from a bunch of the water, sent the water away. This is a concentrating effect, right? So we know that XW should be larger. Okay, so that's kind of some of these principles that we're dealing with here. All right, so how do we manage it then? Well, one of the key terms is called the food to mass ratio. We kind of talked about this in a way when we were describing microbial growth in, let's say, a batch system. Like generically, they can grow some speed, right? So if they're growing some speed, they're really happy if they have lots of food compared to how much microbes they have. That's what this term is getting at. This F to M ratio, this is just going to be one term, by the way, so do not treat this as a division or a fraction. This is just the F to M ratio. That's how the book is writing it, so we are terming it. This is one term. This is one variable. Okay, this is not two variables. Say just one variable. So do not try to multiply both sides by M, for example, or anything like that. Right? Just keep it S/M. I'm just going to follow the book's terminologies and warn you that could get confusing. Okay, so we have an equation for this food to mass ratio, and it will be intuitive once we break it apart, but it equals Q times S naught divided by V times X. The units are going to be the food compared to the mass, right? But this is actually on a daily basis, so it's a little more than just a ratio. It's a daily ratio kind of a thing. So it's the milligrams of BOD per milligrams of VSS per day. So on a daily basis, what amount of food are we adding compared to the amount of mass that we have? So that'll make sense when we see the flow rate. That's going to give us the per time times the substrate concentration. So volume per time times mass per volume gives us mass, right? If we just follow the units there, that gives us the mass with a per time at the bottom. And then below that, we have V times X. That's a volume times a mass per volume. It gives us mass, right? So that's how we end up getting deriving our equation there. Pretty straightforward. It's the daily amount of food we're adding normalized by the amount of bacteria we have there. And if we recall that mu, that specific growth rate constant, our rate constant for how fast the microbes are growing, that depends on how much food we have. It's really actually depends kind of on the food to mass ratio, right? It's how much food is available to the bacteria. So that's why this F/M, this food to mass ratio, is critical. And in general, we want to keep it in a range that keeps our bacteria happy. There are some bacteria that grow better if sometimes they starve and sometimes they have food. There are some bacteria that are designed for different environmental conditions, essentially. You could think about it. So bacteria in our guts, they get kind of mad at us if they're hungry. So some of them will start signaling, and your brain will tell you you're hungry. But it's actually partly your microbe signaling to you. Just like, hey, we're mad. Why don't you feed us more carbs and stuff, right? So that's literally a thing that happens in our own guts. In an environmental system like a wastewater treatment plant, we're generally growing those types of bacteria that just like to continuously eat, right? So it would actually be important or potentially disruptive if we suddenly had no wastewater come in for a few days. That could actually be a problem for a wastewater treatment facility, because you might get bacteria that are happier with other conditions or could outcompete these rapidly growing ones under those conditions, and maybe they don't sink as well, right? Because if they start floating, you get some conditions that you get a different type of bacteria growing, maybe the food changes. You could have sludge flotation, and then your clarifier doesn't work, and then you're not recycling your sludge, and everything kind of breaks down. So if you have sludge escaping, bad news, because A, you've got a ton of sludge just being discharged, and B, you're not having your system recharged with extra bacteria, and so not only are you discharging a bunch of sludge, but before long, you are also now not getting very good treatment in here, and so you are discharging a bunch of high waste load plus sludge, right? So this becomes a big problem pretty quick. So if you ever pass a wastewater treatment plant and it smells awful, something like that might be happening, because normally for this this type of aerated system, it won't smell great, but it won't smell terrible. With all that aeration, it's going to keep it oxic. It's going to keep the smell kind of just smelling a little bit like dirt, and maybe hints of sewage, right? Because you're at a sewer plant. But it shouldn't smell all that terrible. But if you have this flotation system, then downstream or in the clarifier, you're probably going to be having oxygen issues in addition to everything else. So it just gets bad pretty quick. So keeping a good food to mass ratio, therefore, is important. That's one of the parameters that operators will aim to control. And you can control that if you can control how much mass of bacteria you have. So if you have a different amount of food coming in, well, maybe you should adapt how much bacteria you have inside the tank. That comes back again to controlling that waste activated sludge line. Okay, so more details on that. We've covered hydraulic retention time quite a bit. And if you look at the hydraulic retention time in this system, it doesn't change if we add the recycle line. Because what we're going to do is, again, we're treating this as 0 in terms of effective volume of where the reactions can occur. So we're kind of neglecting any volume with a clarifier. Then when we add the recycle line, the overall V over Q for the system is still V over Q when we add the recycle. So when I say the hydraulic retention time, HRT, does not change, adding that recycle line, even though we're increasing the flow through that particular reactor, we are also recycling that flow through the reactor, right? So the residence time doesn't change. That hopefully will make sense if you draw the control boundary outside of that tank, outside of the recycle. You'll notice that it's just going to be in there for however long it's going to be in there. You could recycle at any rate here, and the overall time will stay the same. Okay. All right, so that brings us to, well, what does change? That should be the solids retention time. So this is a concept we've not talked about yet, but you'll see, I mean, in this picture, you can kind of see, I think this is a recycle return for the sludge. So you see this very nice brown slurry there, just coming right back into the tank. And so when we talk about solids retention, or sometimes we'll call it a cell retention time, I think the book has it that way, we will write it theta C for the retention time of the cells, the bacteria cells that we're trying to grow. This will change when we add the solid retention time. And unless, if we are recycling, these are not the same. So in the pond example, the solids retention time should be the same as the hydraulic retention time. There's some bacteria cells growing and they're leaving, right? It's the same speed at which anything else is happening. But when we harvest these solids and recycle a portion of them, now we have a difference. This difference is going to help us control that food to mass ratio like we're just talking about. So this will be important with how much mass of our bacteria are going to be in our system. So let's find this cell retention time. It's not too difficult. So if we say theta C, if you think about it, we should have some amount of cells in the entire volume, and we need to look at how quickly they're going through a system, right? Because theta, if theta is equal to V over Q, then the cell retention time should be something like the volume times the amount of bacteria divided by Q times the amount of bacteria, right? It should take kind of that form, right? It's a per time unit, but instead of the water that's going through, we're talking about the microbes that are going through, right? So given that, we can write that as V times X divided by both flows that are leaving our system, because we have a flow here and a flow here that are crossing our control boundary. So with that, we can say it should be Q in the effluent times the X in the effluent plus Q in this waste stream times X in the waste stream. But we recall this should go to 0 because XE should be 0 if we're operating well. So we're assuming we are. Then our cell retention time simply becomes Vx divided by QWXW. And notice that is very similar to V over Q, right? It's almost theta. It's almost our hydraulic retention time. But we have the X here, and we have to plot where X is leaving. The fact that we are getting nothing to leave in the effluent simplifies this and gives us just this equation. So this is also on your formula sheet. I think the food to mass ratio one is, this one is, so here, if we need to know something about, our QW, and we know maybe perhaps by design we need some cell retention time, that's an easy way to find our QW. Likewise, if we need to change our theta C, QW is a good thing to change. So taking a step back for a moment, if you look at what we can control in this system, the past couple of equations, the food to mass ratio, can we control Q in a real practical system? And I'm asking you, do you think we can control Q? Let's say we have a domestic wastewater treatment plant. We tell people stop flushing. Okay, can't do that. Can we? I mean, I guess maybe we could have an equalization basin or something to limit it if it goes really high, but in general, we can't control it too well, right? Because we tell people to stop flushing their toilets, and that's not going to work out so well, right? Okay, what about S naught? Can we control the incoming wastewater strength? Yeah, probably not, right? Maybe we could try to tell the sky to stop raining and diluting it, or again, people to stop flushing so much solids or whatever. Probably not a whole lot of control there. Maybe, again, a stabilization pond or something. How about the volume of our reactor? Not on the, I mean, maybe we could have one offline so we can go to that if we had to, but that seems like a lot of trouble. Probably not on the fly, right? Not something we can adjust. So that the food to mass ratio, the one thing that's remaining that might be adjustable would be the X, okay? We look at our cell retention time. V is, again, out of the question. X, yeah, that's kind of a maybe. We should be able to control that in some manner. The XW, that's going to depend on X. And the QW is where we have a control knob. And so given some cell retention time that we want to have, to give us a particular X, our control knob becomes QW. So given some operating conditions that we need to maintain, the theta C, we can change QW to adapt X and XW there. So that's really kind of the lever we use to control wastewater systems in general. Okay, so I've got an example problem for us to work on, and this is a bit of a comparison I think I gave this as an exam problem at some point. I don't think I stopped and decided to just keep it as a lecture problem instead. But this kind of gives us a comparison between the a non-recycle and a recycle system. So I might not go through every part of it here, but I'm putting it in these slides for you to have some practice where you see fit. So we'll talk through it, and I'll give you, I'll have you do pieces of it. We'll work those together, and I'll probably skip some pieces and tell you, hey, if you wanted more practice on this, like the example problem we did at the start of this problem at the end of last wastewater lecture, you know, this would be a good point to work on it. And I think I have those solutions that I'll post that for you as well. Okay, so this says, consider a wastewater treatment plant that's being renovated to include a return activated sludge line. So assume the parameter, the influent MLVSS concentration is 0 without the recycle line. Following parameters are known about the reactor, and it acts as a CSTR. So we have a reactor here, a bunch of parameters. We have a design that we're supposed to have a solids retention time of 8 days, like we were just talking about. Okay, so we've got a bunch of parameters. And then part A says draw a schematic of an activated sludge system before and after the addition of a recycle line. So take a moment and draw that. So in both cases, we can probably imagine there's going to be some sort of a clarifier before. It's not too relevant. Probably all right if you didn't draw that. But then also in both cases, we're going to need a clarifier afterwards. That's part of the design. We need something to separate our microbes back out. It's part of why we're growing them in the first place, to catch all those, do that separation. So in both cases, we should have a clarifier. Water is going to leave that clarifier, and we will have a waste line from those clarifiers. So the difference will be in the return activated sludge line, we have a recycle line here, right? So that's what I'm referring to when I'm saying we're going to recycle. Now, in both of these cases, this X naught was 0, and the X naught in this case would be before the addition of that line. So I'm not sure if it's this problem, but sometimes, you know, it might be worth thinking about, okay, well, once you merge the recycle line with the influent line there, the primary effluent, what is the new, like, I don't know, X naught prime, right? You'd have something like that, whatever you want to call it, that's actually entering the tank. You might be interested to like find that concentration, for example. And that, I don't know, that might be something we're going to do in a moment. Okay, so then part B says, what is the final substrate concentration in the no recycle system before the RAS line is added? So this is going to be asking what is, that's not what it's asking. What is the final substrate concentration in the no recycle system. So that with the no recycle system, that should act a lot like that pond, right? We have 0 microbes coming in, we have a CSTR, and that's about it, right? So this is the same problem essentially we did, some different numbers, parameters are a little different, but in general, this is going to act like the pond example. So I'm going to let you do that on your own as you see fit. Okay, so then part C, again, imitating that other problem. What is the removal efficiency? That would be S naught minus S divided by S times 100%, right? So you could take your answer from part B and solve that. Part D is, again, following that same deal. What is the X in the case where we had the no recycle system? Okay, so if you wanted some practice, each of those pieces you can work on your own. See if you can kind of replicate what we did in class for the that example problem. All right, then. So part E, we can put to practice some of what we learned today. So this one says if the sludge has a concentration of 12,500 milligrams VSS per liter, which is now recycled with a new recycle line at a flow rate of 150 cubic meters per day, what is the resulting, and I'm saying quote influent, so now we had 0 influent, but now we recycle, so we have a new like X naught prime kind of thing. So draw that up to make sure you understand where it's going. That's entering the tank. I give you the hint to draw it up and don't overthink it. So take a couple minutes on that. This is the type of problem that in the past I've given on an exam and people way overthought it. All right, so hopefully you've had a chance to think about how you approach this problem. And if you pay good attention to your diagram and kind of write things up there, what you'll notice is you're kind of just doing a mass balance on this little point right here. You've got the recycle line coming in. You've got some amount of bacteria in that recycle line. That's XR. We know what that is. We have a Q coming in. That's the Q for the entire plant. And we have an influent concentration of microbes at 0 at that point. Xe is 0. And then we have some Q that's going into the reactor. I just labeled that Q in. Maybe there's a better way to say that. And I went ahead and said, well, that's also X in, and the problem is really asking us for what is X in. So to solve this, we just recognize that Q times Xe is going to be, sorry, is that Xe? I'm labeling this as Xe today. I guess that's the primary effluent. Not to be confused with Xe over here, sorry. So I'm just gonna, I guess I'll relabel that. Let's say X naught. So let's say Qx naught is going to be equal to, let's see, that's one of the inputs, but we actually have two inputs into this little juncture, right? If we're doing a mass balance on that juncture, then the other one is Q with a recycle times X in the recycle. The combination of those two should give us the Q in times X in when we're solving for that in factor. Does that make sense? Any of you try to do something a little fancier with the other equations? Guess because I warned you, warned you not to. So you might think to yourself, oh shoot, I have to solve for something using theta C or something, right? But just take a look at your figure first to see if you can do something simpler like this. So X naught goes to 0, right? So this goes to 0. And so effectively what we have here is when we want to find X in, that's simply going to be QR XR divided by Q in. We can also write Q in as in terms of knowns, so this will be QR XR divided by QR plus Q. So that would give us a way to kind of put everything into known values. So this here, now we probably ought to put this 150 cubic meters per day into cubic meters per second or vice versa. So let's get a calculator. I guess we'll take the 0.01 cubic meters per second and multiply that by 86,400 seconds in a day. So we'll say Q is that many cubic meters per day. Okay, so then we can solve this problem. We'll say QR, that was 150, times the XR was 12,500, or yeah. And then we'll divide that by the 150 plus 864. And that should give us a value here for the X going into this tank at 1,850. Right? So X 1849 milligrams VSS per liter. Now, one thing we could check here is just to double check ourselves, that should definitely be lower than the 12,000, right? Yeah.
The units are different.
Units are different. Did I mess up the
Yeah, QR is at meters cubed. X is at liters.
You're correct. So, but in this case, it's okay. The reason is because I want I want the final units to be in liters. Sometimes when we're just finding a concentration like that, as long as the flow rates here are the same, they're going to cancel. And so our final concentration will be okay. So I could convert it to cubic meters, but there's no need to because I'm not merging it with those units. It's kind of staying separate. But good catch there. So if you did solve this per cubic meter, it's not typical for us to solve that, but that would be 1.849, right? We divide that by 1,000 for that conversion. All right. So the next problem says, in the new system with the recycle line, the resulting food to mass ratio is 0.58 milligrams BOD per milligrams VSS per day. And so what is the new MLVSS in milligrams per liter? So this is saying what is the X new once we installed the system and now we were given this particular parameter, right? So we're told the food to mass ratio is 0.58 milligrams BOD per milligrams VSS per day. Okay, so we'll need the equation here. And if you recall, we said that this is, well, I mean, we can take a look at the units, right? The amount of food per amount of bacteria per day. So we had a flow rate times the inflowing amount of food divided by, I think it was the volume times X, I think. I'll double check that, but I think that was how we derived that. So take a minute and solve that part. Yeah. That equation is fine. Okay, so it turns out this is not too complicated of a task if we recognize that X that we're looking for is kind of in the definition of the food to mass ratio. So if we know the food to mass ratio, or we're given that here, we can solve for X by saying that's going to be the volume, which was 550 cubic meters, times that substrate concentration. That was given here, 220 milligrams BOD per liter. And again, we'll watch this to make sure that the units come out properly. What am I doing? Volume was on the bottom. Sorry. Flow rate, 0.01 cubic meters per second. Volume is on the bottom, 500,000 cubic meters. And then this food to mass ratio was given in milligrams BOD per milligrams VSS per day. So again, we have an issue where we have the flow rate in seconds, but we need it in days. So we did that a minute ago. That was 864 cubic meters per day. So we can solve for that. And sorry, I should have written the actual number here, 0.58. Milligrams BOD per milligrams VSS per day. So we should be able to just solve that if we have, we'll notice we've got cubic meters and cubic meters. We've got seconds, so if we put that in days, we're okay. Days and days. The milligrams BOD should cancel, and we should be left with milligrams VSS that comes to the top per liter as a units for X. So anybody have that completed already? Should be 655. Yeah. Yes. Thank you. So we're going to take this and multiply by 500 to fix that, divide by 550. So 596, that sounds more correct. Sometimes I have these numbers and I'm like, that's, but 596 definitely sounds like the right one. All right, thanks for catching that. All right, make sense? Okay, I think this is the last part. So we say, what is the flow rate for the waste activated sludge line given these conditions? So given all that, what is QW? Right, and we just solved for X, 596. And earlier we were given the XR was 12,500. To where? This one? Units of what? I think I inserted this number for, yeah. So this should be swapped there. Exactly right. It should be. All right, so if we take a look, we should be able to recognize that our theta C equation is going to be pretty helpful here. That's really the one place we find QW. And if you kind of look backwards through these calculations, we can kind of see that this control over the theta C using QW kind of points to each of those other pieces, right? Points to the X concentration, points to how much we're actually recycling and all that. So if we put this in, the volume's 550 cubic meters, 596 milligrams VSS per liter, and then we have that cell retention time of 8 days, and we have that recycled concentration of 12,500. All that together should give us a final waste flow rate of a little over 3 cubic meters per day of sludge. So this is a pretty small flow overall. You know, the reactor itself is only treating 0.01 cubic meters per second. That's a pretty small flow rate compared to, you know, maybe most municipal plants, but it's an example here. One thing you'll notice is as we get into dealing with the recycled system, we have several equations here that kind of control or point to important parameters that are involved, but we're kind of moving away from solving an actual rate equation inside a mass balance because, you know, it can potentially get too complicated, but also this type of control we have over it is kind of simplified. This recycling of that waste is very convenient for control over wastewater. It really helps that way. All right, so a couple quick announcements. Okay, sorry, there's one last little question. I'll say that the hydraulic retention time before and after does not change, right? Theta is equal to theta, is not necessarily equal to theta C, right? So a couple announcements. Do be checking the Moodle page for quizzes. Again, I think I have one or two due by the end of tomorrow, for example. So be looking at that. There should be those this week and another after Thanksgiving. So I'll close with that and say see you on Tuesday.